All right, thank you very much. So our talk is entitled Automated Adipocyte Analysis, Quantification of Adipocyte Size, Lipid Droplet Distribution, and Total Lipid Content for Evaluating Potential Anti-Obesity Therapeutics. I think that's the best title yet. So I should note that for the context of this, this project, we kind of fabricated a company named Adipolyze. It's not a real company that we have founded, but we thought for framing the video and demonstrating the commercial potential of our research, kind of framing it in the context of a fake company might be interesting. So it's not a real company yet, maybe. So I don't think I need to motivate why we're interested in obesity. Professor Lee did a great job of that, and it's always in the news. We know that obesity is a problem. What I do want to talk about is why we're studying fat tissue. Typically, when you think about targeting obesity, you think diet and exercise. And fat tissue has typically been thought of as this inert storage depot, which stores energy in the form of triglycerides and then mobilizes energy when it's needed. However, recent emerging data indicates that adipose tissue is an active endocrine organ which responds to signals from other tissue types and actually secretes signals of its own. So this is a very familiar slide, which you saw just a few minutes ago. So I just want to give you a brief overview of adipose tissue biology for those of you who might not be experts in the audience. So as already mentioned, we start with the stem cell population, which then commits to the preadipocytes. These preadipocytes can either divide and proliferate into more preadipocytes, or they can differentiate and become mature or immature adipocytes. Once they differentiate, they can no longer proliferate and divide. So our focus is on this hypertrophic growth, which is the lipid accumulation. So as we mentioned, the fat cells store fat as visible lipid droplets, and that's how they become hypertrophic. So there are many, there's a huge metabolic network within the fat cells, and there are many targets which we can potentially look at to reduce fat accumulation in the cells. So I'll come back to this a little bit further, but the point of the slide is that there are many targets, and we're looking at many targets, and we need a good system to evaluate these targets, and we also need a good program to analyze the effects of these targets. So the first step was establishing a system in which we could grow the fat cells and understand how they're developing. So typically in the lab, we grow cells on flat tissue culture plastic, which you can imagine is not very representative of what's going on in the body. I don't know of any flat, hard plastic surface in my body. I hope you don't have any in yours. So we moved towards a 3D system where the cells are actually embedded throughout a collagen matrix. This is mimicking the extracellular matrix within the body. And you can see from this top image of tissue culture plastic to the second image of 3D collagen culture, we see much larger lipid droplets within the cells, and we think this is more representative of what's happening in vivo. In addition to that, we know that fat tissue isn't composed of just fat cells by themselves, but there are other cell types incorporated as well, and the main other cell type is endothelial cells, which make up the vascularization. So if we incorporate endothelial cells into this culture model, we also see a huge increase in the triglyceride accumulation. So all of this is just kind of trying to develop a model system which more closely resembles the in vivo environment. So what we needed was a non-invasive method to analyze these culture conditions. And as was already mentioned, analyzing the lipid droplets is a very attractive option. However, sitting in front of a computer nights on end, manually counting and identifying cells is no fun for anyone. So that's kind of the motivation behind this project, is we wanted a non-invasive, automated, regular, non-biased method to analyze these images. So I'll turn it over. Mic change.
Actually, you can use that.
All right,
Go
great.
for it.
Just hold it.
Right here. Yes, okay. All right, so yeah, like James said, we really were looking for a non-invasive method to analyze these, because beforehand, obviously we want to quantify the reduction of lipid, the hopefully reduction of lipid from one experimental condition to the next. And before we used to do that using a triglyceride assay. So that involves physically breaking apart all of the cells and measuring by a chemical reaction how much triglyceride was in the entire sample. And that works really well. It gets you really good results, but like Professor Lee said, it takes a long time. It costs a lot. And also, one important thing that hasn't been mentioned yet is you lose all information about the size distribution of the fat droplets, which is extremely important in some of James's experiments with Perilipin knockdown. So image analysis was a really good tool to use, and we used it for a while even before making these programs. The problem with it was that it was all completely manual. You literally used to open up 50 or so of these images from an experiment and click and drag across each of these fat droplets, and you'd get a number. You get the diameter, you enter it into an Excel spreadsheet. You go to the next, the next, the next. There's a lot. So, and there's probably 50 images per experiment. So we would really like a way to not do that anymore. So the first thing we did was looked in the literature to see what other people had done, because we imagine we can't be the only people trying to do this. So this first slide here, or this first set of images here, this is what we found a paper that converted these these grayscale images to black and white images as a way to identify where the fat was. And they set just a fixed threshold value where everything above the threshold value gets converted to white, and everything below the threshold lightness gets converted to dark. So this evaluates this image based on the one characteristic that these lipid droplets tend to be the lightest parts of the image. And as you can see, it doesn't do a really great job. It comes up with a lot of false positives. There's a lot of speckles in here. It turns out that their images were a little bit darker than ours, so there's a lot of white in this image that really isn't fat at all. So we said, okay, let's try adjusting that threshold a little bit. And that's what we did here. We adjusted the threshold, and we could get better results, fewer false positives, but we're also missing a lot. You can see we're missing a lot from here to here. So when I look at it, any of these images, the way I would describe these fat droplets would be they're light, they're circular, and they're surrounded by a dark boundary, and that dark boundary is also circular. So rather than just qualifying based on one characteristic, I wrote a program that uses all four of those characteristics. So the first thing you do here is you choose a low threshold. Oh, go to the high threshold is easier to understand. First, you choose a high threshold so that most of the pixels are qualified as dark, and only the very brightest ones are qualified as light. And that way, I can find all of the light lipid droplets and then also a bunch of white noise. Then I do the same thing, go back to the original image and choose a very low threshold, so most of the image would become white, and only these dark rings and some dark noise are qualified as dark. And by inverting that and filling in the holes, I can get this image, which is now, since I inverted it, is white only at the darkest parts of the image. And by intersecting the two, I can get an image that includes only the light objects that are surrounded by dark boundaries. And from there, I wanted to take into account the fact that they're so circular. So a circle has the highest area to perimeter ratio of any two-dimensional shape. And I used that fact to write a circle finding algorithm that finds the most circular objects in this image and the most circular objects in this image and adds those. And then this is actually sort of a whole nother program that Esha did. She worked on it, she's not here. This is a lot like what Professor Miller was talking about. She uses a random forest classifier. So basically, you just load a bunch of these images into MATLAB, and Esha manually clicked through and identified pixels as either background pixels, boundary pixels, light interior pixels, or dark interior pixels. And then after you train the program gets good at recognizing those pixels and classifies them, just like Professor Miller was talking about before, into those categories. And this program does a very good job. The only disadvantage of it is you need to select the cells beforehand, whereas with the first program, you don't need to select the cells. But here, the results are much better, and the correlation is excellent.
Okay, so we have this great program which can analyze where the fat droplets are in an image, but how do we actually use that program and what results can we actually get? So here's that slide I was talking about again. And I just want to highlight pyruvate carboxylase and perilipin, which are the two enzymes which we're looking at right now. Pyruvate carboxylase catalyzes the reaction from pyruvate to oxaloacetate, which is a very important precursor in fatty acid synthesis, which then is synthesized into triglycerides. Perilipin is a lipid droplet associated protein which sits on the outside of the droplets. And in its activated state, it shields the droplets and prevents them from being degraded by enzymes within the cell. And it also helps to form larger droplets within the cell. So both of these enzymes are very important in triglyceride synthesis and triglyceride storage. So if we use siRNA to knock these enzymes down, first off, we see a decrease in the lipid in the cell, and this is through our enzymatic assays. But what's important is that these enzymatic assays actually correlate well with the image data that we get. So on the right up here, I guess you can't really see the laser. On the right, you see the droplet area, and this is averaged from 8 images per condition, and you see a significant decrease in the average droplet area. You also see a redistribution from large droplets down to smaller droplets upon treatment with anti-perilipin siRNA. So we see the results that we expect, and we can actually get these results without having to disrupt the cells. So data correlates well with enzymatic assays, so in the future we won't have to do those assays. Furthermore, for pyruvate carboxylase knockdown, we also see about a 30% reduction in triglyceride accumulation, and again, it correlates really well with the image analysis data. And in this case, we analyze kind of the total lipid amount in an entire image, average that for several images, and we see a significant decrease. So just to kind of summarize, we know that knockdown of perilipin and pyruvate carboxylase both reduce triglyceride accumulation, and perilipin knockdown leads to a redistribution in the droplet size. And we used image analysis to evaluate these knockdowns in a non-invasive way. And we can analyze adiposides not only in our own lab, but we're working with several collaborators who are looking at similar and actually very different problems. Our research is applicable to academic labs, possibly pharmaceutical companies, and even the healthcare industry in general. So that's kind of the commercial application of our research. So we'd just like to acknowledge past and present group members. Specifically, Ning Lai did a lot of the prior work of my research. A special thanks to our advisors and to Stephen and Geraldine Ricci for this wonderful opportunity.
Questions from the audience? For the professor too?
Great.
You wait long enough.
I have one, not the specifics. But did the prize contribute to the learning exercise? Did it raise the intensity or maybe the interest in the project?
Oh, absolutely. I think it definitely encouraged us to, we were already talking about working together, but it really brought up about the, the interdisciplinary aspects, and also trying to figure out how, this kind of very academic research might be applied on the broader sense. I think a lot of times in academic research we tend to focus on this one particular problem, and it helped us to kind of think about, well, is there anyone else out there who might be interested in what we're doing and might be interested in it on the commercial scale.
Thank you. By the way, I'm Bill Messner, the chair of mechanical engineering. Looking forward to meeting you. You're using this siRNA to knock down stuff, right?
Mm-hmm.
And so do you see that might actually be a therapy? I mean, siRNA's been around. People have been talking about it as a potential therapy, but is there a way to use it as a therapy?
The difficulty with siRNA is the delivery. It's very rapidly degraded in the body, and that's because your body has natural defense mechanisms to prevent foreign genetic material from getting into the cells. So there's been a lot of work and a lot of different companies looking at how to actually deliver that. And I think there's been a lot of recent breakthroughs. In the context of our research, we're kind of more looking at it just as an investigatory tool for right now, and hopefully in the future with research that's going on industrially, the delivery will be figured out as well.
Thank you.
No other questions? So we have the team has already received their cash prize. but we have some certificates, and I thought I would bring Steve up as well, and maybe we can get a picture. So there's one for your teammate. So we can open it up maybe, and maybe we can, you want to stand?
Let me say a couple of things. First of all, I see all the talent in this room. And the only way I'm going to be associated with a prize is I'm giving it. I was a very ordinary student at Tufts, but the platform Tufts gives you, the opportunity to work at something that interests you, creates enormous opportunities. But over my career, and the interest in the Interdisciplinary prize, it's been the intersections of markets, the intersections of disciplines, the intersections of ways of thinking that have created the opportunities. And that's why I had interest in getting behind this, or trying to get behind it. And I think it was an exceptional case for me in terms of when it came to that. So whatever I'm supposed to do with
So,
it.
well, congratulations, Chris.
Thank you.
And,